Hi, we are back. All right, we left off on prophase 1. We're in meiosis 1. And the wonderful thing about prophase 1 is how the homologous chromosomes inside every nucleus within a pre-gamete cell, because all these cells are going to become gamete cells eventually, how these homologous chromosomes find each other and then cross over. And in the process of crossing over, this is when they start to begin to exchange genetic material with each other, which is really, really cool. So in order to be a homologous chromosome, it has to be chromosome 1 from your mom's side, chromosome 1 from your dad's side. 1 and 3 wouldn't be homologous. So they don't find each other, they don't mix and match. But the 1s find each other, the 2s find each other. So in this beginning example for meiosis, we're starting with 4 chromosomes, 2 1s and 2 2s. So that's the end of prophase 1. We have crossing over. The point of crossing over is called the chiasmata, and once they cross over, they form this new structure called a tetrad. So let's move on to, after prophase 1, let's move on to the next phase, which is metaphase 1. So I'm just going to keep drawing them back and forth, and then, like I said before, we're going to write it all out. So metaphase 1, here's what's going on in metaphase 1. So once the cell finishes undergoing prophase 1, the nuclear envelope is breaking down, all of that. By the time we enter metaphase 1, the nuclear envelope is completely gone, and we still have our duplicated centromere. And if you remember back in mitosis, the chromosomes, which were all 46, were lining up along their centromere, along the middle. In this case, it's a little bit different in meiosis. We still have a metaphase plate like we did in meiosis or in mitosis, but in this case, the tetrads are going to line up along the metaphase plate. So compare this to mitosis. Back in the somatic cells, you would have 46 chromosomes lining up along the metaphase plate. In meiosis, you have 23 tetrads lining up along the metaphase plate. So there's still 46 chromosomes, but they're now buckled together through crossing over. So you have 2 chromosomes basically mushed into like one tetrad that's going on here. So the tetrads are lining up along the metaphase plate. And this is a super important part of genetic variability. The first part of genetic variability, the genetic variability that you see, is the crossing over event right here. This is huge. So crossing over, this whole process allows for the DNA from your mom's chromosome and the DNA from your dad's chromosome to start exchanging material. Metaphase 1 also allows a big genetic variability component to happen. And during metaphase 1, this is something that happens. It's called independent assortment. And here's what that means. I'm going to actually show you. So if I take the tetrads of chromosome 1 and I put them up here, who's to say that in this metaphase plate, this is going to be the site where we start to see the first split. So just like in mitosis, where we lined up all the chromosomes and one chromatid went here, one chromatid went here, we're going to see something very similar in meiosis. But during metaphase 1, these tetrads are so interlocked together because they're crossed over each other. Who's to say that all the mom chromosomes have to go on this side and all the father chromosomes have to go on this side? There's no rhyme or reason to how this actually happens. So you're going to get all these tetrads lining up along the metaphase plate, but you have no idea who's going to go on what side of the metaphase plate. And that makes a huge difference because where they line up, they're going to stay, and that's when they're going to split again. Because I want you to be able to think about what's going to happen in our anaphase 1. So we're going to just arrange these completely independently, and I'm going to put chromosome 1 for mom here, chromosome 2 for mom here, chromosome 1 from dad here, and chromosome 2 from dad here. So this is the 1s, this is the 2, here's 1 and here's 2. So the tetrads are lining up along the metaphase plate, but this time they're lining up along their chiasmata, the point where they cross over. So They're lining up, didn't really show it as well, but they're lining up along the chiasmata. And of course, you have kinetochore microtubules again. But there's something different this time about the kinetochore microtubules. It's only on one side of the tetrad. So it's not, see how it's only on this side but not on that side? Only, it's on the outermost side of each tetrad. And that's super important. And you also have your non-kinetochore microtubules that are also lengthening the cell like they have in the past for mitosis. So you got kinetochore microtubules on either side of the tetrad this time, and they're connecting. And this is super important that this happens because remember, these are going to become your future egg or sperm cells. You need the separation to be good. If I don't have this microtubule in place, let's just erase this one. If I don't have that microtubule in place, hopefully you can see what's going to happen next. When these chromosomes start to migrate to the poles, nothing is going to pull this one over. This whole thing is going to go over to one side, and that cannot be good. So we want these kinetochore microtubules to actually connect to both sides of the tetrad. But these ones on the center part really don't get touched, which is fine. That's actually what you want to happen. So during metaphase 1, the tetrads now line up along the metaphase plate. So you would actually see 23 of these. And there's still 46 chromosomes because we have 1, 2, 3, 4 chromosomes, but we have 2 tetrads. Okay, so that's metaphase 1. Let's keep moving. I'm just going to go back and forth on the board here. So we have prophase 1, metaphase 1, and now I'm just going to erase prophase 1 and show you anaphase 1. If I had a longer board, I would just continue going all the way down, but I don't. And hopefully, on your paper, you can make this work. So if we go this direction now. So for anaphase 1, you can start to imagine how this is all going to start turning out. I have my centrosomes. And now, thanks to the independent assortment, this huge, genetic variability event that happens within meiosis. So we have crossing over, we have independent assortment, meaning we have no clue on how these tetrads are going to line up along the metaphase plate. It plays a big deal now because we're going to start to get to our first split of meiosis. So just like mitosis, when we split apart the sister chromatids, we're going to take apart the tetrads now. And here's what happens. Let's just focus on this top tetrad right here. When this chromosome gets pulled over, it's going to come over as sister chromatids, but now they're not identical sister chromatids anymore. Because of this crossing over event, we're actually going to be switching out genetic material here. And the tips here that are crossing over with the green chromosome are now, we're going to have a green tip chromosome here, and on this green one, we're going to have a black tip chromosome here on that one. So I'm going to draw these out and I'm going to fill in the colored parts that got swapped just to really heighten what's happening here. So let's draw this one up here first. So once they get, the tetrads are going to split and the non-identical sister chromatids now are going to start migrating to the poles. So this black one is going to come over here and this green one is going to come over here. But thanks to that crossing over event, I now have green on both sides here, and this one has swapped out black on that side. Same thing's going to happen over here. So that black one is going to go over here, and this green one's going to go over here, but I'm actually going to get green here and black here. So these sister chromatids, they're not identical anymore. I now have genetic information from that homologous chromosome on here, and vice versa. They swapped genetic material. And of course we have our kinetochore microtubules over here. We're lengthening out the cell, and there we go. So that's anaphase 1. It's very similar but very different at the same time, because now what we have done is we've separated the tetrads, and our chromosomes are now non-identical now at this point. This chromatid does not match this chromatid anymore. We have non-identical sister chromatids. All right, let's get to telophase 1. So I'm going to erase metaphase 1. I'm just going to go back and forth. So now, you can kind of guess what's going to happen here. This cell is going to split right here. So this is the moment the cell becomes a haploid cell. At this point in the game, it's diploid. I have 2 1s and 2 2s. Now when I go to split this cell, I'm only going to have 1 1 and 1 2, 1 1 and 1 2 in the other cell. So we're creating a haploid cell. We're creating 2 haploid cells at this point. So for step 4 in meiosis 1 is telophase 1 and cytokinesis, actually. We're going to lump it all together. But we don't call it cytokinesis 1, just telophase 1 assumes cytokinesis happens, obviously. And so we're going to create 2 cells here, and I'm going to make them a little bit smaller so I could fit them in here. So here's 1 cell, and here's the other cell. So if I split this right down the middle and create 2 new cells, here's what's going to happen. I'm going to have my centromere over here, and my centrosome, forgive me, over there. And so in this cell, I'm going to have these 2 chromosomes right here. I'm going to have my black one and my green one, but along with their exchanged material on here. So I'm going to have this guy, and I'm going to have this guy, but I'm going to carry on some of that. And then same thing on this side. I'm going to have my green one and my black one. And I'm just filling in where they exchanged, just so it really highlights it for you guys. Okay. Now, if we were to number them, this would be chromosome 1, and this would be chromosome 2, this would be 2, and this would be 1. So now, after the end of telophase 1, we have essentially, we created 2 haploid cells, daughter cells, made of chromosomes with non-identical sister chromatins. So these are, they're not twin sisters anymore, they're non-identical sisters. So made of chromosomes with non-identical sister chromatids. That's the whole point of telophase 1. Okay, we're not done yet. We got to do meiosis 2. But from here on out, it actually resembles a lot of mitosis. There's no more crossing over, there's no more independent assortment, it's just happening, it's doing its thing. Technically, I guess you could still say there's independent assortment during metaphase, because you don't know which way these are going to line up. But there's no more crossing over, there's no more major genetic variation events that are happening within the cell. So let's just keep this up for now, and let's run through what we just talked about with prophase 1 and metaphase 1 and anaphase 1. Let's write this stuff out, and I'll keep this over here because this is where we'll start up for the next part for meiosis 2. Hopefully this is helpful. It always helps to see what we're talking about before you write it out sometimes, because it would just be a bunch of words and you couldn't visualize what we're talking about. So if for 1 was prophase 1, here's what's going on during prophase 1. The big thing that's happening is it's a combination of prophase and prometaphase during mitosis, if you will, because the nuclear envelope is breaking down. Nuclear envelope breaks down. Microtubules begin to grow from the centromeres, or centrosomes, forgive me, microtubules. From the centrosomes. And then importantly, and this is the big genetic variability part, crossing over occurs between homologous chromosomes. Change ink for you guys really quickly. And what I want you to know, the chiasmata is the point of crossing over. So you could have multiple chiasmata along homologous chromosomes. The way I drew it was super duper simple. I want you to actually imagining that both X's are on are totally around each other. That's what's actually happening. So point of crossing over. Oops. CHI. And now the crossed over, the homologous chromosomes are now called tetrads. So the crossed over chromosomes are called tetrads. So you got a couple vocabulary words here: chiasmata, you got your homologous chromosomes, you got crossing over. Those are the new vocab stuff we were talking about. Crossing over is just essentially the point where they do cross over and you're exchanging genetic information in between all those crossing over events. It's really, really cool. All right, so that's prophase 1. Let's describe metaphase 1. So metaphase 1 the tetrads line up along the metaphase plate by their chiasmata. And here's the important genetic variability part. The tetrads independently assort. You have no idea which way they're going to assort. It's independent. They're not dependent on anything, so they just assort independently. Independently assort along the metaphase plate. That's a big deal. All right. And now for anaphase 1. It's a whole lot easier to go through it now that you know what it looks like. So for anaphase 1, number 3, anaphase 1, the tetrads separate and essentially the non-identical sister chromatid chromosomes migrate to opposite ends of, or migrate to opposite poles. little bit quicker in meiosis because you know what's happening already. It's the same mechanisms and machinery that's being used in mitosis, same proteins on the kinetochores that are doing all this. That in metaphase 1, you had all those kinetochore microtubules, same things going on. It's just that the whole purpose of what's happening with the chromosomes is super important during meiosis. And then number 4, it's already on the board here, telophase 1 and cytokinesis. Essentially, the cell, the cytokinesis part, you get the cleavage furrow, you get everything that you did in mitosis. Same thing. The cell splits, and you don't even bother rebuilding the nuclear envelope. We're just going to keep going. Like you do rebuild the nuclear envelope, but then it disintegrates again quickly again. So it comes and goes. And then eventually, you make 2 haploid daughter cells made of chromosomes with non-identical chromatids. And that is the end of meiosis 1. Come back, I know this is a little bit quick because you kind of know the details of what's happening. When we come back, we're going to do meiosis 2. We're going to do it very similarly, where we're going to start where we left off with telophase 1. And when we enter meiosis 2, we're going to start with prophase 2. And you're going to see how from here on out, it's just going to go pretty straightforward. It's the same thing, but it's the very last step. You're going to see, oh, this is why we're doing everything the way we are. So come back. We're going to start here. I'm going to keep this part of the board up so we can continue our story for meiosis 2. I will see you for prophase 2.